What happened?
It's okay.
Oh, there you go. Our opening keynote speaker, Dr. Professor Libby Hausrath, who will speak to us today. A very exciting talk. I'm really looking forward to it. Around planetary science, astrobiology research, and the use of core facilities. Thank you so much for being here, Libby. I'm going to give a quick overview and talk about your background. So, Dr. Libby Hausrath attended Brown University, where she received her bachelor's in geology and chemistry with honors, magna cum laude. She received her PhD from Penn State University in geoscience and astrobiology, where she was an she received her PhD from Penn State in geoscience and astrobiology, where she was an NSF graduate fellow working with Dr. Suzanne Brantley, studying basalt weathering on Earth and on Mars. She received a NASA postdoctoral fellowship to work at NASA Johnson Space Center with Dr. Douglas Ming, where she began to study phosphate mobility on Mars. She became an assistant professor at UNLV in 2009 and was tenured and promoted to associate professor in 2015, promoted to full professor in 2021. At UNLV, together with her graduate students and postdoctoral scholars, she studies geochemical interactions on Earth and on Mars. She and her students are funded to use a combination of laboratory experiments, fieldwork, and geochemical modeling to understand geochemical interactions. Her research group has been funded by NASA for over 14 years. She's a participating scientist on the Mars Science Laboratory Curiosity rover, and she has been funded by NASA to be one of 15 scientists in the Returned Sample Science Participating Scientist Program on the Mars 2020 Perseverance rover. As part of this work, she is working with the entire Mars 2020 science team to help select samples for return to Earth. She has also recently been selected for the Mars Sample Return Measurement Definition Team. This is an international team of scientists who work to determine the scientific measurements to be made on Mars samples upon their return to Earth. These samples will be the first sampled materials from Mars ever returned to Earth and have the potential to transform our understanding of that planet. I give you Dr. Libby Hausrath.
This is just the pointer. Okay, great, thank you. Thank you very much for that kind introduction and for the opportunity to talk to you all about the exciting things that are happening in Mars science these days. All right. Okay, so to give you an outline of what to expect, I'll first give you an introduction to Mars and the history of Mars exploration. Then I'll talk to you specifically about the Mars 2020 Perseverance rover and Mars sample return, and then our work on using transcriptomics to support future human exploration of Mars. And then there should be plenty of time for questions. All right, so Mars has fascinated people for millennia, and one of the reasons for this is that Mars is in many ways very similar to Earth. It's quite similar in size, so you can see the schematic here. Mars is about a third the size of Earth, and then you can also see the relative size of the moon. So because it is smaller, it has less gravity, so about 62.5% gravity on Mars than on Earth. And it's much farther from the sun. And so the average distance from the sun to Earth is about 93 million miles, and the average distance from the sun to Mars is about 142 million miles. So farther, much farther from the sun and less light. The atmosphere of Mars is much less dense. The atmosphere on Earth is about 100 times denser than on Mars. And our atmosphere is, of course, primarily nitrogen and oxygen. And the atmosphere on Mars is 96% carbon dioxide. So a very thin atmosphere, but primarily carbon dioxide. And then because it is so much farther from the sun, Mars is much colder than Earth. So the average temperature on Earth is 57 degrees Fahrenheit with a range from minus 126 degrees Fahrenheit to 136 degrees Fahrenheit. Mars instead has an average temperature of minus 81 degrees Fahrenheit, with the coldest being minus 284 and the warmest being 86 degrees Fahrenheit. And so the length of the Martian day is also very similar to the length of a day on Earth. So a Martian solar day is called a sol, and it is 24 hours and 39 minutes long. So a little bit longer than our day, which means that when we're trying to work on Mars time, we get a little bit offset, a little bit more offset each day, which is why when Mars missions start, people work around the clock to be able to work on Mars time. And so now looking at history, Mariner 4 did a flyby in 1965. So this was the first close-up image of Mars, and it showed the presence of craters, of volcanoes, of much more dynamic and diverse surface than anyone had expected. And so it really changed the impression of Mars from that mission. Then Viking 1 and 2 were the first landers on Mars that returned data, and that occurred in 1976. And so you can see the image here of Viking 1 digging trenches on the surface. And this was really exciting because the atmospheric measurements from Viking helped understand that Mars meteorites were in fact from Mars. And also it contributed a lot of excitement to the search for potential past life on that planet. Sojourner was the first rover on Mars in 1997. So you can see the image of Sojourner here. It had an instrument for collecting chemical data. It also was able to take a lot of pictures. And it was supposed to have a 7-day mission. It actually operated for 83 days. It was a technology demonstration, being the first rover. And one of the really exciting things that it showed was very rounded pebbles, showing that there had been enough water to polish these pebbles and make them smooth and round. And then from Sojourner in 1997, we have the Mars exploration rovers in 2004. So these were Spirit and Opportunity that landed in very different places on the surface of Mars. And these collected a lot of data and images and sent those back to Earth and operated for an incredibly long time with from a planned mission of 90 days. Spirit operated for 6 years and Opportunity for 14.5 years. So they vastly exceeded expectations. And one of the really exciting contributions that they made was the evidence that there was past liquid water. Liquid water is very important for habitability, so finding this past liquid water was very exciting for thinking about Mars as a potentially habitable environment. So this is an image here showing the big contributions of the MER rovers being the presence of past liquid water and carbonates, showing where the carbon dioxide from a thicker, warmer past atmosphere could have been removed from the atmosphere and stored in rocks. So then, in terms of orbiters, the Mars Express in 2003 and the Mars Reconnaissance Orbiters in 2004 collected data from around the planet on Mars. So by orbiting, they can collect chemical and mineralogical data of the surface, and that is the only data that we have for most of Mars. And the really exciting observations made by these orbiters were the presence of clay minerals. So this is an image from Mars Express here, and the blue on the surface indicates where clay minerals are. And clay minerals are very exciting because they form in the presence of liquid water. So again, this is evidence of past liquid water and potential habitability of Mars. Then in 2008, the Phoenix lander landed, and so on the left, you can see the image, the picture of the Phoenix lander after it landed, and then the digging in the soil that Phoenix did there. And the really exciting discoveries by Phoenix were, again, carbonates. So again, evidence that carbon dioxide could have left the atmosphere and been stored in minerals. And then also the presence of perchlorates. And so perchlorates are a very strong oxidant, and so finding them on the surface tells us more about what the surface of Mars is like. And this was an observation that nobody even knew to look for. There was no idea that there were perchlorates until it was measured that they were there. And then in 2012, the MSL Curiosity landed, and that's an artist's rendition that you can see on the screen there. So this is really amazing that in 15 years, we've gone from Sojourner, a tiny technology demonstration, to MSL Curiosity, which is the size of a small car. And so this shows the relative sizes of the models of Sojourner, the MER rovers, and MSL Curiosity. And so MSL Curiosity was so heavy that it had to be landed on Mars in a different way. And so this is an artist's rendition showing a landing of MSL Curiosity, and this is called the Sky Train. So you can see that the lander is gently lowering the rover down onto the surface of Mars, which of course is incredibly terrifying, but it landed successfully. And it landed in Gale Crater. So this is an image here of Gale Crater. And the interest, Gale Crater was selected as a landing site because it was a past lake. And there are sediments in the bottom of this lake that could have been a habitable environment. And so this, again, is of great interest in this progression from Sojourner, which observed this evidence of water, the MER rovers, whose goal was to follow the water, and then MSL Curiosity, whose goal was to assess the evidence for potential habitability. And so habitability is generally considered to be the presence of sufficient raw materials, so carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur, sufficient energy, either from light for photosynthesis or from chemical, for example, redox gradients, liquid water, which is essential for all life on Earth, and then sufficiently favorable conditions, so pH, temperature, and salinity. And so Gale Crater was found to have all of these and considered to be a habitable environment. And so this is the next step from following the water to a specifically habitable environment. So then from MSL Curiosity, we move on to Mars 2020. And so you can see artist's rendition of Mars 2020 next to MSL Curiosity. You can see that they look very similar. The rovers are very similar with some improvements like better wheels to protect from the rough Martian surface and different instruments, but very similar rover bodies. And so the science objectives of the Perseverance rover are to examine the geology, so to better understand Mars as a planet, astrobiology, to look for evidence of ancient microbial life, to cache samples for return to Earth, and to prepare for human exploration of Mars. So Perseverance launched July 30th in 2020. And this was, as I'm sure you remember, in the middle of the global pandemic. And so just an amazing feat by the engineers to be able to finish this project and get it safely launched in the middle of the pandemic. So here it's launching from Cape Canaveral. And then it cruised through space for more than 292 million miles and almost 7 months. And then it landed in Jezero Crater on February 18th, 2021. And so Mars 2020 was able to use a new landing technique called terrain relative navigation. And so it takes between 5 and 20 minutes for a signal to go between Earth and Mars. So we can't actually drive or land the rover. And so in this case, the rover was for the first time, was able to take descent photos as it is landing, compare that to an orbital map, and then divert if necessary. And so the rover is doing this all autonomously. while it's landing. And this is really exciting because it allowed the rover to land much closer to this sort of dangerous fan that was of great interest, but that it couldn't land on. So it was able to land much closer to that than previous landers or rovers had been able to. And so the Mars 2020 rover again landed by the sky crane. So you can see the artist's rendition of it being landed here on the surface. And then this is an actual picture from the sky train looking down on the rover being landed, which is just, again, just incredible that that's possible. And then here's an image you can see where Perseverance landed, and then where the parachute and back shell and the descent stage and the heat shield were all discarded far enough away to not injure the rover at all. And so this was the first image that the rover sent back. And so this was from when the rover had landed and was able to send this back. We knew that it was safe on the surface and that it was communicating with Earth. So this was a very, very exciting image for everyone to see. And there you can see the landing site, Octavie Butler landing site, marked with the yellow star there. And the reason Jezero Crater was selected is because, like Gale Crater, it's a crater lake, and it has both an inlet and an outlet. So that indicates that it was full enough of water in the past to fill up to that outlet. So this was very exciting in terms of, again, looking for a potentially habitable environment and assessing possible evidence of past life. So this is the rover's traverse so far. So it landed down on the floor of Jezero Crater, and then it traversed across that floor and then up onto the fan, which is sediment deposited by liquid water. And then the little blue rover at the end of that is where the rover is now. And it has come out of the crater and is now on the crater rim. And so that URL on the bottom shows where the rover is, and so that's, anybody can look at that to see where it is. The blue, the little blue with the helicopter shows where the helicopter is, and then each of the little red balloons shows where samples have been collected. So Perseverance has 23 cameras and 7 scientific instruments. So it has 9 engineering cameras. So the HAZ cams help look out for hazards in the front and rear of the rover. The nav cams help navigate. And then there are science cameras associated with the different instruments, as well as cameras associated with entry, descent, and landing of the rover. And so these are some of the instruments. So the SuperCam instrument uses LIBS, laser-induced breakdown spectroscopy, to measure the chemistry of rocks. And so you can see this artist's rendition here of SuperCam shooting a laser at a rock. And this allows us to have chemistry, mineralogy, and also measurements of organic matter if it's present from a distance, which is very helpful as the rover is traversing through the environment. Sherlock and Pixel are proximity instruments. They're present on the arm of the rover, and they can measure the chemistry and mineralogy, as well as potential organics if they're present, in very tiny areas with very high resolution. So combined with SuperCam, this allows us to look at the very fine scale measurements of what's present in these rocks. Perseverance can also look below the surface, so it has an in situ radar to measure below the surface, and this is very helpful in terms of analyzing what's at the surface to understand and see what is going on beneath the surface. And then, like Curiosity, Perseverance also has its own weather station. So it can measure wind speed and direction, temperature and humidity, and the amount and size of dust particles. So this is very important for preparing for potential human exploration to understand the characteristics of the modern surface. And then also preparing for human exploration, Perseverance has a tool to convert carbon dioxide in Mars's atmosphere. Again, the atmosphere is 96% CO2 to oxygen, and this is MOXIE. And so Perseverance tells us about what it finds through the Deep Space Network. So there are 3 locations with large communications antennae that can receive signals in California, Spain, and Australia. And so Perseverance sends data back to Earth's deep space network via the Mars orbiters. So there are satellites orbiting around Mars, collecting data, and also they collect the data from Perseverance and send it back to us here on Earth. And so, as I said, it takes about 5 to 20 minutes for a signal to travel between Earth and Mars, depending on where the planets are in their orbits. And so everything that the rovers do is sent up to them in a plan for each day that they then autonomously carry out. And so it might take pictures, it might use its laser or drill into a rock for a sample or drive. All of that is in the plan that goes up to the rover each day. And so this is sort of looking at a day or a sol, so a Martian day, a little bit longer than an Earth day, in the life of a rover. And so the rover wakes up at the start of the sol around Earthrise, and that is when it gets its plan, the files that it needs in order to execute the next sol's plan sent directly to the rover from Earth. So that tells the rover what it's going to be doing that next day. And so the rover carries out the sequences commanded in the plan. So it might drive or drill or collect data. So you can see here it's put out its arm to measure a rock. And then when the afternoon orbiter overflight approaches, so you can see the streak in the sky, which is the orbiter satellite that is collecting the data from the rover. Then the rover sends some of the collected data to the orbiter, depending on the, ordered by the urgency of the data. So data that are needed immediately for the next day's plan, so images of where the rover is going to drive, or perhaps rocks that we want to drill or analyze are sent back first. And then data that aren't going to be used in immediate planning can be sent back later to the orbiter and then to Earth. And so the orbiter records the rover data and then relays it back to Earth. And then the rover continues to do its plan, moving through the day. And then it can work a little bit at night, but it needs to recharge its batteries during the night. So it also needs to sleep to recharge the batteries. And then it wakes up in the early morning to send data to the orbiter overflight. And then the orbiter records the rover data and relays it back to Earth. And so some of the things some of the data that the rover can send back to Earth includes the latest weather. So this is from a particular sol showing a high of minus 8 degrees Fahrenheit and a low of minus 109 degrees Fahrenheit. It can send back information about Martian dust. So this is showing the wind blowing the Mars dust across the surface of Mars. It can send back acoustic information. So for the first time, we can hear things on Mars. So record the wind as well as the sound of the laser shots, which provides some more information about what the target is. information about MOXIE. So MOXIE successfully pulled in the CO2 and electrochemically split it to form oxygen. So you can see on the plot there, time on the x-axis and total oxygen produced on the y-axis, and it successfully produced oxygen. So this technology demonstration in preparation for future human exploration. And then, of course, the Ingenuity, the helicopter. And so Ingenuity was supposed to perform 5 flights over 30 days, and it performed 72 flights over 3 years. And so it did a great job. And so here's an image here of Ingenuity flying. And then this is a map showing, hopefully this will work, showing the all of Ingenuity's flights, and so the distance that it makes. You can see from where we landed, it started flying and then crossing across the crater floor. So we went around to look at, basically this direction. Then we went back up towards the fan to look. So those are water-deposited sediments. So we wanted to go up onto to the fan. So then the Ingenuity crossed the dunes, went up onto these sediments deposited in the past by water. And then you can see that it continues to make its way up from the front of the fan up over the top of the fan here, and then eventually completing its 72 flights here. And so it flew over 10 miles, and for a very long flight duration. And so this was an incredible technology demonstration, and also really exciting to be able to see the information that, whoops, I don't know what we have going on here. See the Ingenuity, what Ingenuity was able to do. And then eventually it had a hard landing, which damaged one of its wings, so it couldn't fly anymore. And so then we also had, this was the first abrasion on Mars. So on the rover, we're able to abrade the surface to be able to see beneath the surface. So the surface might have coatings or weathering rinds, and so to see the rock beneath that, we can, abrade into that. So this is the first abrasion. And then this is showing the multi-instrument analysis of the sample. And so the different instruments, the red and blue and yellow, are the footprints of the different instruments. And that is able to give us very detailed information about the mineralogy and chemistry of the rock. This is the first Mars rock sampled in tube 266. So on the right there, you can see a picture on Earth of the sample tubes that were made and then are on the rover, and where the rock samples can be sealed into those tubes for return to Earth. And then so far we have 27 samples and 3 witness tubes collected. And so this is the map showing the abrasion for each of those samples. And so the witness tubes are empty tubes collected to look for any possible contamination, so that when the samples are analyzed, we can look at any background for the samples as well. And then in addition to rock, we also have Mars soil. So this is an image taken after the rover collected 2 samples of soil, which were then also sealed into the tubes for return to Earth. And then, so in addition to all the rock samples and the soil samples, one sample has been very exciting recently, and that's Chayava Falls. And that's because, so if you see in the red stripe in between the 2 white stripes, you can see the white and black spots, which are being called leopard spots, and then the little black dots, which are being called poppy seeds. And so the leopard spots and poppy seeds, the dark part of those is phosphate, and that's exciting because phosphate is essential for all known life, and that because there's also been organic matter definitively detected by the Raman in this sample. And so in terms of looking for potential past habitability or assessing the possible evidence for past life, this sample is really exciting for being able to look at these, look at the phosphate and organic matter further. to Chayeva Falls is really exciting. And so the samples that we'll get back from this collection will be the first samples collected on Mars. We have Martian meteorites, and so these are images of shergottites, knocklights, and chassignites, which are different types of Mars meteorites, from my colleague at UNLV, Dr. Arya Udry. And so they've told us a lot about the history and past of Mars, but they weren't collected by us. They're random samples that just happen to be ejected by an impact off of Mars and land on Earth. And so the samples that we're getting from Mars are collected in situ and have very exciting characteristics like the presence of phosphate or organic matter. And so this is an image here. On the left, we have Buzz Aldrin collecting the lunar rocks. And of course, we learned so much from the samples from the moon. They're still being studied to tell us about the moon. And then on the right, we have the artist's rendition of Perseverance collecting a sample. And so this is just a schematic showing the plan for the cache. So as we drive along, we have a plan for collecting samples from the crater floor, samples from the fan, samples from outside the crater rim outside Jezero, for the Regulus sample. So all of these are being collected with a goal of being able to examine different past environments on Mars. And then they're being cached. And so this is a schematic showing caching on Mars. And so far, one cache has been deposited on the surface of Mars at Three Forks, and the rest of the tubes are being carried on the rover, and with the plan to deposit them for collection to return to Earth. The initial cache is essentially an insurance plan in case anything happens to the rover. And so this is the first cache. You can see the 10 tubes there that have all been deposited on the surface of Mars in a flat spot where a lander could come and retrieve them. And then the remainder, more of the tubes, remain on the rover. And so this is a notional 3-mission Mars sample return campaign. So we are at the sample collection part of this by Mars 2020. So Mars 2020 is driving around, looking at all kinds of interesting rocks. They're being drilled and sealed in tubes for collection. The next step is to collect them and return them using the Mars Ascent Vehicle, the MAV, which launches the orbiting samples, the Oz. And then the Mars orbiter captures the Oz and brings it back to Earth. And so Mars sample return can help enable future human exploration of Mars. So this is an artist's rendition of a potential future human astronaut on Mars. And so the samples can tell us lots of things that we probably don't even know about Mars that will be very important for future astronauts. For example, on the moon, the lunar regolith, or soil, turned out to be much sharper than anyone had anticipated. And so it was actually cutting holes in the astronauts' spacesuits, and nobody knew that was going to happen, and it was kind of a problem. And so, getting samples back will help us understand potential both resources and potential hazards for future human exploration, particularly the soil, the Mars soil sample. And so now I'm going to talk just a little bit about some transcriptomic work to support human exploration of Mars. And so in the plan for potential future human exploration of Mars, one idea, this is an artist's rendition, one idea is to use flexible greenhouses to grow plants for both oxygen production and food production. And if they're flexible, then it takes less mass and difficult, weight to build them. And so a flexible greenhouse similar to an astronaut spacesuit, could hold a pressure of about 300 millibars. And so we wanted to know whether algae could grow under these low pressure conditions to produce oxygen and food for potential future human exploration. And so we grew Dunaliella salina and Chloromonas brevispina under low pressure and normal conditions, and this work is being done by Dr. Lena Sissel, who's an assistant research professor in our group and actually here at this meeting. So I know she's looking forward to talking to many of you. And so Dunaliella salina is a halophile, so it lives under very salty, briny conditions, and so that is very relevant to a dry, salty Mars. And Chloromonas brevispina is a snow algae that grows under very cold conditions in low nutrient environments. So again, an extremophile that is potentially relevant to Martian conditions. So we grew these under low pressure conditions, 160 millibars CO2 atmosphere, so representative of what you would get if you condense the current Mars atmosphere to 160, pressurized it to 160 millibars to grow the algae, and then compared that to 1,000 millibars, so Earth's atmosphere, normal atmosphere, and these were performed in triplicate. So hopefully this video will work. Yeah. So this is the low pressure chamber here. You can see that we pump that down 3 times. So we pump out all the air, pump in CO2, then pump it down again 3 times until we get the 160 millibars. Oops. I'll just show that again. 160 millibars atmosphere. Oh, no. Oh, maybe we don't get to see it again. But it was boiling, so, oh, there we go. Yeah, so you can see it boiling. So this is at room temperature, and the pressure is low enough that it is boiling at room temperature, and then we put it in the cold room, and it stopped boiling for the snow algae, and they grew great. And so it's really amazing that they can grow under these conditions that they have not evolved to grow under on Earth at all. And so we were curious what genes they're expressing in order to be able to do this. And so we performed transcriptomic analyses with the Nevada Bioinformatics Center, so a core center at the University of Nevada, Reno. And on the top, you can see we did PCA analysis, and with our triplicates, the large difference between is between our conditions and our experiments, so that's good. And you can see on the bottom the volcano plot showing the upregulated genes in red, the downregulated genes in blue, and then little change in black. And then when we look at the specific genes between these, we can see that Dunaliella salina expressed many more upregulated genes than Chloromonas brevispina. So on the right, we have Dunaliella salina. On the left, we have Chloromonas brevispina, and you can see that largely the Dunaliella salina responded by upregulating, and Chloromonas brevispina by downregulating its genes, and with fairly different responses. So this is something that we're still looking into, but in general, they showed an upregulation of ribosomal RNA processing genes with 50 and 64 transcripts upregulated, respectively, and then they showed very different responses to the conditions. And so a lot more work is needed to better understand the genetic or the transcriptomic responses of these algae for use in potential biological life support systems. And so with that, I would like to just wrap up that Mars 2020 helps tell us about the history and origins of Mars with these samples. It provides information on possible locations that could have harbored water in the past. We're collecting samples for return to Earth. This provides a great opportunity for students and future scientists, so I hope that any students and future scientists will persevere in science and keep going in that field, and also helps pave the way for future human missions to Mars. And with that, I'd like to acknowledge the Mars 2020 Participating Scientist Program, as well as the entire work by the Mars 2020 science and engineering team that makes this all possible. And with that, I'd like to thank you for your attention, and I would be happy to take any questions.
If anybody has questions, please come to the microphones here in the front of the room.
Hi. What happens to the carbon when the carbon dioxide is split up?
Sorry, can you repeat that?
When you make the oxygen, what happens to the carbon?
Oh, you know, actually, I'm not sure. I'm not sure what MOXIE does with that. I should look into that. But yeah, so yeah, sorry.
Very nice talk, thank you.
Thank you.
So in terms of studying organisms on Earth that we're going to bring to Mars, what are thoughts about using, genome engineering to alter these organisms to help us in terms of dealing with things like cosmic radiation and harsh conditions that these organisms are going to have to grow when they get to Mars?
Yeah, that's a great point. Certainly there are very harsh conditions on Mars. And so, yeah, examining what kinds of genetic modification might make it easier is certainly a promising field.
What's the chemical, analytical technique for Pixel and Sherlock?
Yeah, so Pixel is an X-ray, basically an XRF, and so it measures the X-rays of the elements from different depths, and so it can basically gives an elemental map of these rocks with a pixel size of about, I think, 100 microns. And so that's been very exciting for looking at being able to identify different minerals. It also is able to provide some diffraction information as to whether they're X-ray amorphous or not. And then Sherlock is a Raman, and I don't remember the wavelengths for the lasers, but it's basically able to use Raman to identify mineralogy as well as organic compounds.
Hello. I'm wondering about when do we hope the samples will come back? And then if there's any particular sample that you're excited about and why?
Yeah, so the current NASA plan, it has two options that were presented in early January, and so that has arrival as early as 10 years from now, and so that would be very exciting. Of course, everything takes funding, and so it has to be sequentially funded, but that's where it's at right now. So with continued funding, that's where it would be. And I'm very excited about the soil sample. It will be our first sample of soil. Obviously, the meteorites are rocks, and so this sample is our first soil sample. It has samples of dust, which of course is very important for human exploration. It has the potential to interfere with all kinds of human technology, get into equipment and everything. It also has samples of what looks like a global material from all the way around the planet. Rovers and landers in different locations have taken measurements, and it looks very uniform. And so to have a sample of that potentially uniform material is really exciting. And then it also has small pebbles that have been heavily altered by water. And so that's the only sample that, those pebbles are the only sample of material that was too friable to be collected in a drill. So something that is potentially so heavily weathered that it couldn't be drilled. So the soil sample is very exciting. And then Chayava, which has the phosphate and the organic matter, is very exciting for potential examining the evidence for possible past ancient life on Mars.
I'm very interested in those algae producing oxygen. Did you see that they were producing the same amount of oxygen than under normal pressure? And have you seen that they were in constant stress, or were they producing, regulating themselves in a normal way?
Yes, yeah, they were definitely under stress, and they grew more slowly, and they also produced very different amounts of oxygen. So that's interesting. So Dunaliella salina produced much more oxygen than Chloromonas brevispina, and actually, we actually examined a number of other species. Chlorella vulgaris, which is also edible, also produced a lot of oxygen as well, but we weren't able to do the transcriptomic work on that. But yeah, so they're definitely under stress, and but they produce oxygen pretty well.
Yeah, pretty nice. Did they need anything else that may not be available on Mars yet, like microbiological environment or fertilizers, things like that?
Yeah, so there's lots of phosphate on Mars. There's less, or nitrogen might be more of a problem. There doesn't seem to be a lot of available nitrogen, so that would be something that might need to be thought about. Yeah.
Thank you.
How confident or settled is the science around like the existence of large amounts of water on Mars's surface? Is the Mars community pretty confident in that conclusion, or is that still kind of under evaluation?
Yeah, that's a great question. That is generally the consensus view. It is, it's interesting because there's no liquid water on the surface now. But there is a lot of evidence for physical rounding of the rocks, physical transport, chemical interaction, or minerals that result from interaction of liquid water with primary minerals like clay minerals, for example. And there is water ice on Mars, so there's basically a place for the water in addition to escaping into the atmosphere, and nitrogen isotopes suggest about 80% of Mars's atmosphere has escaped into the atmosphere, but there's also water ice, indicating that there is still water on the planet. So yeah, it's certainly there are a lot of things that aren't known specifically about the water that are still areas of research, and Mars is a surprising place. But yeah, that's definitely a consensus view that there was lots of liquid water. Yep.
Okay, thank you. One more question. What are some other cool extremophiles that might be worth considering growing on Mars?
Oh, other extremophiles? Yeah, well, there's been a lot of work looking at, I think, Deinococcus for radiation. Radiation resistance. I'm trying to think what other extremophiles might be useful. But yeah, it's certainly they'll face lots of challenges.
Great talk. So I have a question and a suggestion. The question has to do with the algae. How long did you put it under these conditions? Were you able to see, for example, changes over time through multi-generations?
No, unfortunately, we only have a single time point. I think we grew them, I would have to look back. We grew them until they reached the stationary phase, so that varied in terms of number of weeks, but I think up to about 90 days, if I'm remembering. But yeah, we don't have multiple time points. We have multiple time points for oxygen and chlorophyll and other measurements, but not for the transcriptomics.
Was that just a technical limitation, or did they peter out by that time?
We stopped them when they were still stationary. They do carry on pretty well, so they're pretty low maintenance. So we could look at how long, but we didn't stop them because they died. We just stopped them because they reached stationary phase.
Okay, that's good. So the suggestion, I was amazed to hear that you can actually hear the wind on Mars. And I was thinking you should give that to some pop artist and it could become a #1 hit if they put it to a song.
That would be very cool. Yeah,
Thanks.
Thank you.
My question is, if you do discover compounds that support the evidence of ancient life on Mars, how do you then differentiate whether that life was present on Mars or if that compound hijacked a meteorite that hit Mars?
That's a great question. And so, yeah, certainly NASA's come up with something called the ladder of life detection, I think, which basically, if there's something that seems indicative of life, then basically there has to be much more work to examine that potential possible, future definitive detection of life. And so that would be a very interesting thing to look at, certainly. And there's lots of technology development going on right now to try to look at like the MinION, which is being used on the International Space Station to examine transcriptomic, basically in real time, the expression to look at disease-causing organisms, for example. So yeah, basically it would have to then go up a lot of the ladder of life detection to learn a lot more and be much more definitive.
Thank you.
Yeah.
That was awesome. Thank you so much. I was really struck by how much longer all of these rovers lasted than expected. Is this sort of an example I think everybody in the room knows of under-promising and over-delivering, or was there a lot of luck, or was there something different about Mars?
Yeah, let's see. Yeah, I should also mention that the MSL Curiosity is still going strong after 13 years, so hopefully that continues. I think the initial, promises are very realistic, and so there is, they certainly don't overpromise to begin with. Mars is a tough atmosphere, so I think it's very hard on vehicles. It's very cold. There are large swings in temperature. The rocks are very sharp. The rover can slip a lot, so when it's trying to drive, it can slip a lot on the sand. It can get stuck in the sand. I'm trying to think what else. The engineering is just very solid. I just cannot say enough good things about the NASA engineers and engineers at JPL. They just are marvels in being able to do this. They have a test bed at JPL where they have a mock-up of each of the rovers, and so if the rover gets in trouble, then they can basically use the model to try to model on Earth how to get the rover out of trouble up on Mars, so I think that helps. It's also very conservatively programmed, so if the rover encounters something that it thinks might be a threat to itself, it shuts down, and so then it doesn't do anything autonomously, hopefully, that could damage it, and then people can troubleshoot it on the next day. But yeah, everything's this sounds like super obvious, but I had never thought about it before these missions. Everything's really hard on Mars. Like, it's really hard, right? Like, taking samples is hard, taking measurements is hard, driving is hard, everything is hard. And so, yeah, it's just a marvel of engineering.
Thank you.
Okay, wow. We have one more question about something related more to core facilities, and then we'll wrap it up.
Yeah, I'm Scott Ty. So I've been working with JPL for a while on planetary protection operations. And one of the comments I want to do is just kind of bring this back to the cores a little bit, because we've been spending 15 years making reagents that are DNA-free and getting ready for Mars sample return, if you were. And I just thought I'd take the opportunity, because we have a lot of vendors here, right? And I work with them to try and make DNA-free components, so when we actually get these type of samples back, we can actually test them without having so much contaminating background DNA. My question is, and I know there's somebody right after you to talk, but amino acid content of meteorites that come off of Mars, do they have amino acid content for those meteorites that have been recovered?
Sorry, can you repeat the question?
The meteorites that have come from Mars, have we detected amino acids on those particular meteorites?
Gosh, let me think back. So there are certainly anticipated to be organic compounds on the surface of Mars deposited sort of repeatedly by micrometeorites. So material from outer space. And I believe that has amino acids in it, so that we would anticipate that occurring. I can't remember off the top of my head, but I think that would be a likely, certainly that it's likely to be being deposited.
Okay, cool, thank you.
Sure.
All right, another round of applause for Libby. Thank you so much for coming.
Thank you.
Now I'd like to give the floor to Joe Dragavon, who's going to talk a little bit about our international collaborations with international associations. Thank you, Joe.
Hi. Everybody ready for the opening reception? Well, you have to wait. So as ABRF is trying to make sure you get the most bang for your buck, they decided to give you 2 extra brief talks right after this. So this is the first one. So let me see. I can see that. You can see that. Oh, cool. All right. So, hi, I'm Joe Dragomon, ABRF Executive Board member. This is Josh Rappaport from Boston College, not an ABRF Executive Board member. We're going to talk to you today about one of the activities that is going on within the ABRF, and this is the ABRF and CTLS collaboration. We're going to talk about this because ABRF is trying to expand its impact beyond just the standard society, which you know it as now, and reaching out to like-minded societies throughout the country and throughout the world to bring more to you, to enable you to do more. So, Josh, you want to introduce yourself?
Hi, I'm Josh. I'm the Executive Director of Research Infrastructure and Operations at Boston College.
Thanks. Good job, Josh. All right. And like I said, Joe Dragomon, I'm at the University of Colorado, Boulder, where I hold 2 jobs. I run the Advanced Light Microscopy Core within the Biofrontiers Institute, as well as I'm the Director of Core Facilities and Shared Instrumentation for our college. So who are we? We are both light microscopists. We've been doing light microscopy for a long time. Josh is a cell biologist. I'm officially an analytical chemist, which if you don't know what that means, it means you're not good at chemistry and you're not good at engineering, so they put you in between the two. We both have a passion for core facility life. We've been involved with core facilities for an extremely long time now. And though we are both American, leading this very international society, we both have lived and worked in Europe. We both are married to European scientists, right? So you see we're very well qualified. But I have dual citizenship. Yeah, I got this. Thank you. We all had various COVID projects. Many of you are probably educating yourselves and becoming smarter. I got French citizenship. That's what I did during COVID. Exactly. Vive la France. All right. So the core facility world is small. Josh and I both owe a lot to this person. How many of you in this room know Spencer? That's exactly what I thought. So Spencer, unfortunately, suddenly passed earlier this year. I believe everyone here knows about this. Spencer was a lifelong member of ABRF. He had been a member since early 2000s, or mid 2000s, late, early 2010s. He brought the idea to Europe and found like-minded individuals in Europe and started the CTLS, the Core Technologies for Life Sciences. He's also a co-founder of StrataCore. Now, the two of us have a very strong connection with Spencer. Myself, I was actually a postdoc for Spencer for 5.5 years. It's where I learned all about core facilities and how amazing they absolutely are. I was at the very first CTLS meeting and at the setup meetings for that. I can tell you some stories about how that went. Probably not in this public venue, but some of the private stories would be pretty funny. And Josh met Spencer while Josh was a faculty at the University of Birmingham, and they got to know each other because at the meeting, Spencer noticed Josh taking copious notes, and Josh became the secretary for CTLS. So ABRF is, as I was saying before, really trying to expand out its resources and how it can help each and every one of us in this room. And one of this is to form strategic partnerships with like-minded affiliations. So like core facilities, the ABRF is part of a larger network of associations. And to maximize the support and opportunities provided to our members, we're developing these strategic partnerships. And the first one we're really working on, or have been working on for years now, is with the CTLS. Now, there's a lot in common. If you go to our website, you can find this, word mess of what the ABRF and what it is for you. But if you colorize it, you can see some key phrases in there that we can all take away. If you look at CTLSs, very similar word structure. It's natural synergy just waiting for us to actually work together. We have a lot of similarities. We are member-led associations. We focus on core facilities and shared instrumentation. We have a very similar governance structure, and we're open to anyone from anywhere. We do have some key differences. We're very US-centered here at the ABRF, whereas CTLS is very European-centered, technology-focused. That's what the ABRF is, whereas CTLS is more agnostic. It's more administration. We have research groups. They have working groups. Working together actually benefits us both, right? We have a shared mission, right? We can learn from each other. We don't all have to make the same wheel, right? We can make a much better, much rounder wheel by working together, provide growth opportunities for each of us, and overall, we are stronger together. What are we doing, right? We've said lots of words over the years, but we're going to start delivering, and it's important that the ABRF and CTLS deliver for its members. So we have had some key deliverables that have actually happened and are going to be happening within the next year. We have a joint ABRF and CTLS MOU. The members are producing a book centered on core administration, which is coming out eventually. We are going to make a special issue of JBT. That's a joint special issue. And we're actively participating in each other's conferences, working groups, and research groups. These actions will allow us to demonstrate that we are stronger together and actually are producing more for each other. So I'm going to let Josh explain a little bit about the MOU.
That one? Sure. All right. Thanks, Joe. So last year the presidents of ABRF and CTLS signed a memorandum of understanding, which declared a shared mission to share information and resources. You can check out the agreement yourself if you want. There's a lot of detail there, and basically the outputs that Joe is describing and that we're working on come straight out of this document in large part. And this is a living document, so at any time anybody can speak to the association leadership if they see ways that the agreement can be improved upon. So the first thing Joe mentioned was the book. So we have a book contracted through a publisher, and it is a shared collaboration between the two associations, and it is being co-edited by Julie, Andrew, and myself. And this is our sort of basic, fundamental TOC. And we have a lot of people in this room working on it already. We also are looking at JBT as a great resource through which CTLS can contribute, and we have two very active members of CTLS, Ralph and Natalie, who are being added to the JBT editorial board, and there's going to be a special issue focused on the collaboration of ABRF and CTLS. There will be special papers commissioned and reports, including meeting reports from this meeting and the CTLS meeting. I should mention, there's still time if you're interested in any of this, if you're interested in participating in the book, if you're interested in participating in the special issue, reach out to me, reach out to the other editors, reach out to Ron about JPT, any of that stuff. Please, we're looking for volunteers. And Sheena and Andy have already kindly agreed to co-edit this special issue with Ralph and Natalie. And so basically, as Joe mentioned, why are we doing this? It's, to really work together and do more together than we can do separately. And there are other collaborators as well. So there's the Canadian Network of Scientific Platforms, and you can read the others as well. BINA is a big one that a lot of ABRF members I know are involved with as well. And so you want to finish it up?
Thanks, Josh. So just to summarize, so the ABRF and CTLS, we do have this very natural overlap that really just offers a multitude of, I guess that's the theme music playing me off the stage, right? It doesn't show it here anymore. So there's plenty of opportunities for us to overlap and actually produce more together than we can on our own, right? Just like core facilities really are, the sum is greater than the total. The total is greater than the sum of the individual parts. That's what ABRF are with CTLS, right? establishing these strategic partnerships will, benefit our members, and really will just maximize efficiency and reduce duplication, right? And we are starting with discrete deliverables. We really want to put something back in your hands as soon as possible. If we make it tangible, if we make it so you can hold it, you know it's real. Now, it's going to be all electronic, so holding it will sort of, you're going to have to mentally hold it, but you mentally know it's there. So it's fine. So reach out to us if you need. If you have any questions, we'll be around all conference, and I'll be up here probably way too often. All right, thanks. There's one more talk. Yeah.
Okay, hold on for just a few more minutes. I know everybody's excited to go to the reception. I think, Diane, we're going to talk a little bit about the Canadian Society as well.
Okay, thanks everybody. We won't take too much more of your time. I am Diane Miller. I'm part of the Western Association of Core Directors Organizing Committee, part of the CAB, and part of the EB. I'm also Canadian, and it's my pleasure to introduce Brooke Ring, a fellow Canadian who'll be providing some information about the Canadian Network of Scientific Platforms, also known as CNSP. Brooke is the Associate Director of Operations for the Translational Institute of Medicine, or TIME, at an institute at Queen's University in Canada. Brooke is responsible for the largest biomedical core at Queen's, known as the Queen's Cardiopulmonary Unit. Brooke also serves on the executive board of CNSP, and in her spare time, she is pursuing a PhD investigating the best practices and impacts of core facilities in Canada. So welcome, Brooke. I'm looking forward to hearing about this.
Thank you, Diane. And I'm going to keep this so brief, I promise, so that we can all go party later. Well, again, I'm going to set up a timer and everything, just so I don't go on. Okay, let's see if I can get this working. Does that work? Where do I point it?
Nope, you know it's the big one.
The big one.
There's another click.
Oh, this thing. This? What am I even pressing? Let's see. There we go. All right, so I think so my name is Brooke. Thank you for the introduction. I'd like to thank the organizers for including the CNSP and giving us some time to talk. I thought I'd start off with just describing the mission statement of the CNSP, which is to empower Canadian researchers by advancing infrastructure and expertise, driving innovation, fostering collaboration, and accelerating scientific discovery. I think the best way to kind of break this down, is that's pretty wordy, is to describe our 4 pillars of our strategic plan. So advocacy, education, networking, and professional development. And we measure this through scientific excellence, our focus on careers, building a community, and evolution of both core facilities as well as core scientists that work at them. The executive board is made up from a team of people at different levels. They're bench scientists, they're administrators, and they're people that run core facilities. Most of you may know Claire Brown. She's our president and founder of BINA. She sends her regards for not being able to come today. I'm also joined by our vice president, Vidhu Sharma, who's here somewhere. And then we have a treasurer and 3 members to represent the different districts of Canada, Western, Central, and Eastern Canada. So CNSP is not as big as ABRF. Our metrics are about 230 different core facilities across Canada, and we estimate that we represent over $1 billion worth of research infrastructure. However, we've re-examined that number. It's much higher than that, but it's pending. 30,000 researchers and over 10,000 laboratories across Canada. Here you see a picture of our map. With all the provinces across. Each of those circles and the numbers represent the different core facilities across each province. You'll see that the province of Quebec and Ontario are the largest. That's our most populous areas. CNSP is a little different than ABRF. We don't just represent biomedical facilities, but we also represent material sciences, chemistry, and physics. So it's a very diverse group So our method is kind of a spoke and hub model. If you think of CNSP as being the maple leaf in the middle, each of these are our technology nodes or partnerships. So we represent core facilities on a whole, advocacy, professional development, and liaising with the government, while we rely on our partnerships to help kind of divvy up the cause, especially for technical education, which we rely on our partners with. So that's the CNSP is there, the C and the Proteomics Network and Canada Bioimaging, Helium Group, and some neurology groups as well. So what do we do? The screen is so small for me. We make connections, essentially. So as I said, we represent over 200 different core facilities across Canada, and we really connect these core facilities and these people working at them. We have monthly seminars that we put on. We help create professional development workshops. We have training initiatives through our technology nodes, and we help promote those. We resource, so we provide resourcing. We can help you set up workshops. We can help you source some talent and help with some of the financials. But mostly what we do is advocacy. We work with the federal government. We work with provincial governments to help create strategic plans and give recommendations on a national level. And we also give out awards recognizing research excellence. And then we create opportunities for our community to join into the conversation and have their say, usually done through our monthly series, Community Conversations. And finally, what we're working on right now is creating scientific literature about core facilities across Canada and data collection. So I'll just very briefly go over what we've done, what we're doing right now, and what the future looks like. This is a picture that was taken at our most recent symposium. So as I just mentioned, we've gotten involved with the federal government in advocacy and giving them advice on their national and strategic plans. Canada is a lot less provincially focused as it is more nationally focused for looking for solutions. And we're working with our one governing body that kind of does core facilities. So that's called the CFI, the Canadian Foundation for Innovation. And they've created a new stream for funding of just core facilities. Which is new in Canada. It was just introduced last year. We've recently had our seminar, our conference in 2023, and we've done various position papers. Currently, what we're working on right now is our very popular community conversation, which we have every month, where we talk about hot topics, or we invite a technology node or partner to come and talk about what's new and cool happening in their avenue. We're working on our new strategic plan for 2025 and 2030, and we are continuing our advocacy work and active conversations with the federal government about our funding models. So what's upcoming? CNSP Leadership and Management course. We've partnered with Global Bioimaging and Bioimaging North America. They're having their 50th anniversary in Montreal, Quebec. And we're going to be hosting a 3-day Leadership and Management Summit. So we're going to be doing that. And then it's pushed back our annual symposium to 2026. We're going to have that in the summer in Ottawa. And this is the last slide. I'm at 5 minutes and 30 seconds. Finally, I'm here all week. We have a booth out front outside of registration. Me and Vidu, you can come and say hi to us if you want to join our LinkedIn page and see what we have going on. There's a QR code there. We also have cards and brochures if you want to learn more. Come say hi. We're very friendly. And I am also doing a talk on climbing the career ladder on Wednesday at 10:45. If you're interested in learning more about the research landscape in Canada and career pathways, please come. I think that's it. Done.
Okay, that concludes our opening remarks and keynote. Thank you so much for being here. Welcome to ABRF 2025. Welcome to Vegas. Have a blast, learn a lot. I hope you enjoyed the conference, and it is time for the reception. You can find the reception in the exhibit hall just outside here to the right, and I will see you there.